How do testing kits for the coronavirus COVID-19 detect the virus? The test used for COVID-19 is real-time quantitative polymerase chain reaction (RT-qPCR). Now, I realize that is a string of long words, but I'm going to explain it step by step. It is a very common test used for diagnosis and in research laboratories. The first step of the process is to isolate the viral genetic material. For coronaviruses, this is RNA. The COVID-19 tests require the use of a specific commercial kit for collecting the RNA. But, in general, these kits all work on the same basic principle where chemical solutions are used to lyse, break apart cells and viral particles to free the RNA. This mixture is then applied to a column, basically a porous matrix of silica. The mixture is forced through the pores using a centrifuge, spinning the tube at very high speed. Various solutions are then used to wash column, ensuring that the only remaining substance from the cell's viral particles is RNA, and then pure water is added to elute, or release the RNA from the column for further work. For RT-qPCR, the RNA has to be converted to DNA, but this step is pretty simple. There's enzymes which can perform the reaction and it is typically a quick process. Next is the actual RT-qPCR. Fundamentally, the process is the same as any other polymerase chain reaction and mimics the process used by cells to replicate DNA. PCR requires DNA, which we just made from RNA, primers, which I'll explain in a moment, and nucleotides, basically the raw material DNA is made from. DNA is comprised of two strands, which can be separated by heating the sample, also called denaturation. This separation allows primers to then bind. This is the annealing cycle. Primers are small segments of single-stranded DNA that match the genetic sequence of a region of interest. In the case of COVID-19, the primers match a small portion of the genetic sequence of the virus. The design of these primers are essential as they must be highly specific to the viral gene. If the primer matches any region of a human genome, it will bind to that instead. Also, if the primer doesn't match the coronavirus sequence, the test will be negative, even if a patient is infected. The last step of the PCR cycle is elongation, which is where the nucleotides are added to the primer, resulting in a copy of the original sample, but only a copy of the region you care about. In our example, this is a coronavirus gene. What makes RT-qPCR different is that the nucleotides are labeled with a small fluorescent molecule that gets released during the elongation step. When it is released, light is emitted, allowing a detector to read the intensity of the light. The more light, the more DNA is being synthesized. The results in a graph like the image below. Many samples can be tested at once, typically with a 96-well plate, where each well contains a sample. The tests can be classified into two main types: 1. Molecular tests, to detect antigens. 2. Serological tests, to detect antibodies. The traditional molecular test is real-time RT-PCR, reverse transcription, polymerase chain reaction. In addition to PCR, new rapid molecular tests are appearing in recent weeks. Regarding serological tests, to detect antibodies, these tests have the great disadvantage that they are not useful to make diagnoses in the first 12 days of infection and in its equivalent of the first 7 days of the disease. It is a window period broad and corresponding to the most active period of infection and disease. The window period in which no detection occurs during the first 11 to 12 days of infection, which is equivalent to the first 6 to 7 days from the start of the symptoms of the disease, considering an average latency period of 5 days. In the asymptomatic serological tests are positive when they are 13 to 15 days after the start of the infection, so it would not make sense for them to isolate it more than 3 to 5 days, since when they were 15 days old they would be recovered with a history of infection pass. Only if it was identified that the infection occurred less than 14 days ago, would isolate it until it completes 15 to 18 days from the supposed date of infection. The important thing to identify the asymptomatic is to investigate your contacts, search in your work area, where you take your food, at your home and the places you have frequented. There must be an active search for undetected cases. On the other hand, we have that, in the moderate, severe and critical, the PCR and the IgM can remain positive for more than 30 days. These cases must be considered with the diagnosis of persistent infection x SARS-CoV-2. In them it is observed that viral activity persists, with involvement of the lungs and intestines, so they must receive treatment to reduce viral load despite having more than 30 days from the onset of symptoms.